So couple of quick announcements as usual. First one is that this week's seminar in biology is also going to be about viruses. A slightly less well-known virologist will be talking about viral adaptations, mutagens, and biochemical properties of the non-structural proteins involved in resistance to lethal mutagenesis. This is a new postdoctoral fellow in my lab. He's going to be talking about his PhD work, where he looked at through some basic fundamentals of evolution and using viruses to study some of those really big evolutionary questions. I think it should be a really fun seminar. Of course, I hired him, so I'm slightly biased as far as that's concerned. Second thing is the exams are all graded. I have the Scantrons. I haven't alphabetized them yet, so it's going to be really hard to give them back to you unless you know exactly at what sequence you turned in your exam from like the 130 people who did it. So I will have those on Wednesday together with the Scantrons from the first exam if you would like to have those. Second thing about the exam and questions that people have had about that, I'm going to go through the statistics of each of the questions and then revisit a couple of things again in a lecture on Wednesday. So I won't be answering people's specific questions about the exam until then. After that point, more than happy to, and I'll post them on D2L, et cetera. Because at least in theory, this class is about learning the material rather than doing well on the exams, at least in theory. OK, so any questions about that at this point? We'll take a quick look at the breakdown of scores in just a second here. No clicker questions today, so the few of you who are here can all leave. But did want to just briefly mention, again, my normal scoring here. High score was 46, and then 90% of that means an A, et cetera, 85%, and so on and so forth. If you're in this kind of range or lower, it might be a really good idea to talk to me if you haven't already decided that talking to me is not worthwhile. So that's where we stand. I have not gone and checked to see if it was the same person who scored both high scores on the two exams. Again, I haven't looked at that yet. That being said, 46 is 2 higher than the next highest score, which is 2 higher than the next highest score. So the normalization is probably going to be around 42 when we look at the final scores for the class. Oh, and that does remind me, clicker scores have not been updated yet. No, I will be doing those as soon as I input these scores into D2L, so the exam scores, and the up-to-date clicker scores will be going in hopefully today, depending on how many questions people have in office hours. And again, office hours-wise today, I'd prefer not to talk about exam questions until on Wednesday. Yeah?
So sorry, on here it says that it's 39, it's the out of 46 that you just said it's going to be out of 42 potentially, so we don't quite know exactly.
So these are the minimum. If you have one of these scores, for instance, you have a 36, you'll definitely have a B+. You might have an A-, but you'll definitely have a B+. OK. Again, more questions on the exam. Certainly the exam questions per se we'll talk about on Wednesday. So today we're finally, I think finally, getting to the most exciting part of the class, at least in my rather biased opinion, and that is starting to think about regulation. Kind of everything we've talked about so far is really building up to thinking about how genes are turned on or turned off. And when I say genes being turned on or turned off, it's really all the way from what DNA is being transcribed all the way through to actual protein structure and protein function, although also to some extent certainly RNAs as well. So today we'll talk about regulation in general, which is almost all about the DNA. And if we have time at the end, we'll look at a nice parody some friends of mine made about it. It's all about the base rather than all about the DNA. Talk a little bit about differential regulation. The whole take-home message here is really that different cells, and even the same cell under different conditions, is going to be expressing its genes differently. That's really what makes different cells different from each other, what causes the response of any given cell at any given time to any given stimulus. Really, it's a lot about that gene regulation and how that's going on. Since we're almost all about the DNA, a lot of what we're going to talk about today is DNA binding, and literally the regulatory proteins which are binding to DNA, which is, I brought the model again today to talk about that. A couple of important aspects about DNA binding. One is almost all DNA binding proteins are binding as oligomers. That means more than one individual polypeptide. And for the most part, they're dimers, but there's certainly modifications to that. We talked about motifs already before. Now we'll talk about some of those very specific motifs. Most of them are going to be either a DNA binding motif or a multimerization motif, and multimers here is almost always going to be dimers. Because of this, you have this phenomenon called cooperativity, and basically the whole message with cooperativity is when you've got two proteins that are binding together, particularly if they bind together better on DNA, what happens is the response to those proteins binding to DNA is much more like an on-off switch than it has something, a rheostat, where you just slowly turn things up. And that does seem to be very important in terms of gene regulation. It's important that something is on or that something is off. And so those are important aspects of cooperativity. And then we'll talk a little bit about nucleosomes at the end, together with cooperativity and with why eukaryotes are such a pain to work with, at least from the molecular biology point of view. So first, let's talk a little bit about what's going on with these different cells. So this is sort of the extreme that they chose in the textbook, where you have a neuron and a liver cell, both of them to scale. This cell has almost exactly the same DNA complement as this cell. Again, this can be from one individual, but of course this one looks a lot different than this one, and it's also expressing lots of different genes, this cell relative to that cell. I put same in quotes because we all know when you do replication, be about 1 in a billion changes. Each time you replicate, you got 3 billion, so there can be a few differences from one cell to the next. The other thing is in a few cells, particularly immune system cells, we have huge rearrangements that happen. And so not all the cells in your body are going to have exactly the same DNA in them. Again, it's the exception rather than the rule. Lots of proteins that are being expressed here turn out to be exactly the same between these cells, which is interesting. You might expect them to be really different relative to each other, but it turns out that most of the genes that we have that are coding for proteins are expressed all the time. And I may have talked a little bit about constitutively before. Constitutively basically means things are on all the time. So something that's constitutive splicing. These are the exons that are always used. Constitutively expressed, something which is always being expressed. So most proteins are being constitutively expressed. And then, of course, as I mentioned before, you can even in different cell types have different responses to exactly the same signal. It may turn out that one cell you need to produce adrenaline, and the other cell you need to produce glucagon or something else, even based on exactly the same hormone, exactly the same chemical. So a lot of that is going to depend on your gene regulation, which, again, could be a whole course in and of itself. So it's all about the DNA. We talked before about epigenetics. Yes, there are clearly modifications which are not part of the genetic machinery. But what is pretty amazing and still kind of blows me away is that you can take a nucleus from one organism and put it into an unfertilized egg and get that egg to develop into a clone of this organism that you started with in the first place. And so these were the very first experiments. These were done in the 1960s. So this was quite a long time ago that people were doing some of these cloning experiments. Most people, of course, think about Dolly and so on and so forth. We'll get back to that in just a second. But what they did here was literally take nuclei from one species of frog and put it into an egg from a completely different species of frog. And the tadpoles that you got out were of the species of frog that you got these cells from in the first place, rather than the egg that you brought in here, with one big exception. What would that be? Not so much the haploid genome. These are, in fact, already, here it's unfertilized, but here we've got diploids. So the nucleus which comes in is diploid. But what other genetic material are in eggs? Mitochondria are in eggs. Mitochondria all are descended from the maternal line. So here it's actually going to have mitochondria from one species and a nucleus from another species, which is interesting. And it turns out that these tadpoles, here it shows them as being normal. They're not normal by any stretch of the imagination. They can't actually even develop into frogs eventually. Yeah?
The egg was from a different species entirely, or just a different?
A related species, not a completely different species. No, we're not talking science fiction here. Yeah? Okay, so the question here is, sorry, I didn't repeat the previous question, but here, would this be a de novo species? I would certainly call it a GMO, but how are you going to define a species? Is it something which can actually replicate, et cetera? To some extent, horses and donkeys, you make mules. So, it's not that crazy to start thinking about some of these things. Of course, they're sterile, so they can't actually be passed along. But yeah, the species definitions start to get really confusing when you start thinking about some of these things. Turns out that plants are often much better behaved than animals. You can take sections of many different plants, separate those individual cells, and those individual cells will end up giving you, and I don't really like the fruiting body here, but the whole plant you can recreate as a clone. And in fact, people in agriculture have been doing this for probably centuries, if not millennia, basically cloning through this process, a molecular cloning. And it turns out it really is all about the DNA. Of course, what we mostly talk about these days is our friend Dolly up here, the cloned sheep. 2000, basically what they did, and again, this is a figure from the textbook, it's been done with cows, it's been done with cats, it's been done with dogs. There was a report that this had been done with human embryos. That turned out to be a fake. So there's some interesting ethical issues that come up with this as well. But again, the basic idea is you take cells from one particular breed or species, again, depends on how you describe it, and then transfer it into an egg that has been enucleated. It is really important that you take the one nucleus out before you put the other nucleus in. If the two of them are into together, gives you usually a god-awful mess. So there are a couple of other things to bear in mind about this. This set of cells which are being used are epithelial cells from the oviduct, which again, oviduct, they're not eggs, but they're epithelial cells. One of the things about epithelial cells is they're constantly undergoing replication. So number of cells, particularly neurons, they don't continually undergo replication. But epithelial cells do, because they're getting sloughed off and being reused all the time. And if you go back a couple of slides and look at the frog case, exactly the same thing was true there. These were fibroblasts. These are epithelial cells which are constantly undergoing replication. That means they've got active telomeres. They've got a number of aspects to them. So if you just take a random cell in the body and try and do this, it's not going to work. And in fact, it took the people who were trying to do these experiments with sheep about 20 years to actually figure out how to do this. And then there was this, electrical pulse. And apparently you do have to have an electrical pulse. Nobody has any idea why you actually need it. But it causes these cells to form, and then you do get a cloned cell that comes out here. Now, who knows which of these is Dolly? No, actually not. So it turns out the big sheep here is Dolly, and this is her offspring, which gets back to your question about the different species and why the mule doesn't make a good example here, because this is Dolly's offspring. Regular, bred in exactly the normal way with rams, etc. And so the basic idea here is to show that this is a normal sheep that was created here through this cloning process. If you're looking actually at the mother of Dolly, or at least the surrogate mother of Dolly, had a black face. And so it was really easy, in fact, to tell that you'd had this switch that had happened here. This, again, happened with Dolly. Dolly did get a number of diseases that are associated with old age very early, but we're talking about an N of 1. And so what are the distribution, the different frequencies, how likely are some of these things to, in fact, be surviving or not? As I mentioned before, human cells cloned through this particular process. That one paper that was, in fact, published on this was then since retracted because they had, A, some really questionable ethics, and B, actually faked the results. And I won't, if you're interested, I can give you some more information on that. This process is, in fact, called somatic cell nuclear cloning. Everyone calls it cloning. When we think about cloning from a molecular biology point of view, it's really about one specific piece of DNA, which could be as much as a chromosome, could in fact be a whole genome. But it's the DNA that we're talking about making multiple copies of. In this case, it's a whole nucleus which is being transferred. And the nucleus being transferred from a somatic cell. So when people talk about cloning in a lot of these cases, again, it's about transferring the nucleus. It's not about the actual DNA itself. Yeah? Right, so the question here is about the telomeres and the age of the mother versus the age of the sheep. They did test the telomeres very carefully. Turns out, again, partly because these are actively replicating cells from epithelia, the telomeres are pretty long and they've been replicated well. There's active telomerase in those cells. If, however, you take one that didn't have active telomerase, then there's some issues that go along with that. So, yes, there were some telomere shortening. Again, how relative that was to the cloning, I think, is not entirely clear. Okay, so at least the nucleus you can transfer and get clones. And in fact, people have done this now for many, many generations. Anybody know why they picked sheep? Pardon? So sheep are the same as humans. Oh, well, I'm kind of fluffy, I know, but no, the main reason for this is they had, in fact, engineered sheep to produce protein pharmaceuticals in their milk. And doing that process cost them literally millions of dollars. And once you've done that for one particular sheep, you want to be able to maintain that sheep over long periods of time, i.e. clone them. So that way those sheep are in fact producing this. So goats have been cloned that are doing exactly this, and there are a number of now currently marketed pharmaceuticals that are from transgenic goats that are expressing these human proteins in their milk and are cloned for exactly that reason. Okay, so let's talk about some of the genes now. We'll move away from this nuclear cloning process. People are more interested in it. Take cell biology. Ask me again. I can give you lots more information about it. But let's move down a little bit more to the molecular aspect of things and look at just a couple of those examples that I mentioned basically from the first slide. Different cells expressing proteins constitutively or much more specifically. Here's a nice example of a constitutively expressed gene, the beta-actin gene, important for the cytoskeleton. And it's being expressed in all of these different cell types. How do we know that it's being expressed in these different cell types? This is a really powerful technique that's just come along in the last couple of years. It's something called RNA-seq or RNA sequencing. Basically what you do is you get a cell from embryonic stem cell, liver cell, muscle cell, blah, 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 etc., and isolate the RNA from those cells and then sequence the ... out of it. And you literally get millions of sequences of different RNAs. And what you do is just take all of those sequences of the RNAs, you know the human genome, and you just line all those sequences up relative to the genome. And basically, each of these vertical bars here correspond to all of the RNAs that were found that corresponded to that particular part of the genome. It's an extremely powerful way to study gene expression, and all you need to do is know the genome and be able to sequence. And when we get to the end of the class, we'll talk a lot about some of the what's called high-throughput sequencing. Literally getting millions and in some cases billions of sequences is really quite straightforward these days. So what you can do is just line up all of these sequences in your software. And in fact, this is software that's freely available online. Just have to go to the UC Santa Cruz genome browser. In fact, these are data directly from that. And you can see here's a bunch of RNAs that correspond to this exon. Here's a bunch of RNAs that correspond to this exon. And very few RNAs that correspond to the introns. A little bit, but relatively speaking. And this is, in fact, a log scale in terms of all of this. One other interesting thing about this is that this very 5' end, there are also a whole bunch of RNAs that are in here, even though they're not part of exons. But they're still part of the messenger RNAs that are already being made. We'll talk much more about that when we talk about translational regulation later on. Take an example now of a, oh yeah.
When they do that matching, do they have to look for exact matches, or how much of deviation do they have to?
So the question is, when you're doing these RNA-seq experiments, how perfect do you have to match some of those sequences? It varies. Different algorithms that are used, but usually it's 99% plus identity over a particular sequence, and again, it depends on the technology you're using, how long those sequences are. So yeah, there are some differences, and it's also a great way to find single nucleotide polymorphisms, et cetera. It's RNA, and so going from the RNA to DNA, you introduce some mutations as well, so there's a lot of details about the process here. And literally, just computationally, taking millions of sequences, comparing them to billions of base pairs. This is not a straightforward process by any stretch of the imagination. So you can do exactly the same experiment with exactly the same RNA and exactly the same set of sequences, and now align them to other sequences in the genome. And this is the tyrosine aminotransferase gene. It's a metabolic gene, which is critical for liver function, but not important for all these other ones. And here you find nicely all of the exons of this gene only expressed in liver, and just a little bit of background from some of these other cells. But again, it's the same data set. You just took your RNA, sequenced it, looked at it. So this liver cell RNA is exactly the same as this liver cell RNA. It just depends on where you've lined it up in different parts of the genome. The old school way of doing this was called microarrays. We talked a little bit about microarrays. We talked about genome replication, where you have 2 times as much of a particular DNA right next to an origin of replication, as opposed to here. This is now a RNA microarray. Same thing, however, is going on. You've got a microscope slide. On that microscope slide, you've got lots of individual known sequences that are on that particular microscope slide. And then what you do is you take, in this case, prostate cancer cells, lung cancer cells, brain cancer cells, et cetera, and take those and make the RNA from them, hybridize that RNA to everything which is on your microarray slide, and then look and see if one particular gene, each of the rows on here, which are not labeled because there are 1,800 of these different rows, represents a single spot on your microarray, which would be a single gene. If it's red, that means this gene is overexpressed relative to your mean of all of the other cell types that people are looking at. However, you can also see these genes here, a whole block of genes, which are less than the mean. They're underexpressed. And what you see, if you look at lots of prostate cancer cells, there are differences between each one, but the overall pattern is pretty similar. Now if we take a leukemia set of cells, look, these genes are really overexpressed here, but they're underexpressed relative to that. So why is this important? Well, if you happen to have an unknown sample which has particular expression which is going on there, you can compare the pattern of this unknown to all of these other patterns and find out that it's a lung cancer. If, for instance, it's a metastatic cancer, you find a tumor somewhere else, and you can now identify it very clearly just by looking at which RNAs are being turned on. In this case, it's only 1,800 genes. People are now doing it with all 30,000 genes, same kind of thing. Although what's happening is people are moving further and further away from this microarray technology because it depends on having a slide that's something you can hybridize to just sequencing all the RNA and just comparing it to the genome. But again, the same idea. It's patterns, finding different patterns which are there. RNA is great, but most of the time, and certainly for messenger RNA, that's not what's actually giving you the final phenotype. It's the proteins which are there. Looking at proteins is way, way, way harder than looking at nucleotides. Yeah, there are 20 amino acid side chains. There are lots of post-translational modifications that happen. Phosphorylation, acetylation, ubiquitination. These are just a few of the ones that we've talked about so far. One way to look at different proteins is something called two-dimensional gel electrophoresis. Just depends on your molecular weight versus the isoelectric point. So you separate proteins from high to low in terms of their molecular mass, and then acidic to basic here. Isolate proteins from brain, isolate proteins from liver, identify those that are the same, and this automatically, at least if any of you have looked at this already, shows you how challenging this is. So red are the same. Okay, we got these 3 dots here. They're the same. Where are the 3 dots over here that correspond? I guess it must be this one and maybe some of these ones. This red dot here that's supposed to be the same. It's probably this red dot here. It's a lot harder process to try and identify which of these are which here. Each of those individual spots, you can figure out what it is using mass spectrometry, but it's a very convoluted and involved process. And as if that wasn't challenging enough, you have a number of things like this one over here in liver. This one spot here, it's all at the same molecular mass, but has multiple different charges. All those different charges have to do with how many phosphates are attached to it. So it's all just one protein. Just different phosphates which are giving it different kinds of isoelectric points. But you can do these kinds of analyses and see different proteins. Again, all the blue ones here, not expressed at all in brain, well expressed in liver, and piece apart how all of these things come together. This is a much better way to figure out what proteins are being made, because again, we talk about translation. Translation is great because it can amplify whatever messenger RNA you have. Messenger RNA is easy, but a large amount of messenger RNA doesn't necessarily mean you have a lot of protein, and a small amount of messenger RNA doesn't necessarily mean you've got a small amount of protein. And so using these kinds of techniques, actually looking at proteins, even though they're a lot more complicated, are a lot more powerful because they'll give you that relative amount of protein at the end. And that gets to our overall picture of gene regulation, which is basically what we're going to spend almost the rest of the course talking about. The active proteins here at the end, these are what are actually doing the job, most of the jobs. Again, there's some RNAs and some other things in there as well, but for the most part, it's the active proteins that are important for the actual function, most of the cells making those amazing dendrites that you see in neurons, turning on the metabolic genes that you happen to have in the liver cells. So it's really about that active protein. How do you determine how you have an active protein? Well, again, it's all about the DNA, or almost all about the DNA. You need to take that DNA, transcribe it into RNA. This is your primary transcript. That gets modified into a messenger RNA. This messenger RNA gets exported into the cytoplasm. That messenger RNA gets translated. Once you have this translated protein, you often have some kind of control of that activity that can be being put together. It can be being modified, etc., until you finally have this active protein over here. why are we going to spend so much time talking about transcriptional control? Well, the reason we're spending so much time talking about transcriptional control is that this is the first step in the process. Transcriptional control is where you're going to start spending all of the energy to make the RNA, to make the protein, to modify that protein. So if you want to have really fundamental control, energetically most efficient, you're going to have that control here at the transcriptional level. There's a problem, however, with having control at the transcriptional level. It actually takes time to make transcripts, to modify the RNA, to export the RNA, to translate the RNA, etc. So if you want to have a really quick reaction to something, say a hormone or whatever it is, then you're going to want to have regulation happening here at this very final level once you've got protein that you can turn off, turn on, turn off, turn on, etc. So we've already talked about some of these things. These will be phosphorylation, there'll be a GTPase activity, changing a structure of an already present protein. Okay, but let's move back, talk about transcriptional control. We're going to talk about transcriptional control for the rest of today and, in fact, most of the rest of the week. So how do you regulate transcription? Transcription is going from DNA to RNA. Well, you have to then deal something with the DNA. All of these transcription factors, again, factor just means protein. And what they're doing is regulating transcription. This is different than the TFs that we talked about last time. Well, last midterm again. Last time is all like pre-midterm. All of those are their general transcription factors. These are transcription regulatory proteins that are going to act at lots and lots of promoters. In some cases, almost all promoters are going to be interacting with these. On the other hand, the specific transcription factors are the gene regulatory proteins. These are the ones going to be interacting with one or a few genes. Turns out that a large proportion of our genome, if you look at the 20-odd thousand protein coding genes, about 200 of those are specific gene regulatory proteins. And the fact that our genome is spending so much of its, at least, coding space on these proteins means they're probably pretty darn important. All of these transcription factors bind to intact double-stranded DNA, which is, again, why I brought my structure again today. Most of these are going to interact with these so-called cis regulatory sequences. So cis versus trans. Everybody remember cis versus trans from your general chemistry? Cis is right together, trans is far apart. You think about double bonds, cis double bonds and trans double bonds. Exactly the same kind of thing here. So cis is right next to whatever sequence it is that they're regulating. And we'll talk a lot more about that a little bit later on. There are some trans examples as well. But for the most part, they're cis. They're right next to the rest of the gene, and so the regulatory sequences are right next to it. And most of these gene regulatory proteins are very specific in terms of how they bind to the DNA. Well, how do you get specific binding to double-stranded DNA? We talked about this when we talked about double helical structure, the DNA structure way back. So it's mostly about the major groove of DNA. You've got hydrogen bond acceptors, hydrogen bond donors. In the case of AT base pairs, you have methyl groups. And these are chemical structures that can be recognized by, we're talking about factors here, so they're going to be amino acids that are part of proteins. So how do you get these interactions? Through all of the amino acid side chains, but particularly amino acid side chains that are going to be involved in hydrogen bonding. As we just said, there's got hydrogen bond donors and acceptors in your major groove of DNA. What are the major hydrogen bond donors and acceptors in side chains? These are going to be your polar side chains. So they're not the ones that are charged. Those are actually very often going to have the non-specific interactions, say with the charge interactions in the phosphate on the outside of your DNA. But those are going to have hydrogen bond interactions, going to be things like asparagine, arginine. Why did he do this funky capitalization in here? That's because the single letter code for asparagine is N, and the single letter code for arginine is R. I don't expect you to know these. I mentioned amino acids right at the beginning. I would say lysines and arginines, yes. Glutamic acid and aspartic acid, yes. These are just good to know that you've got hydrogen bond donors and acceptors. I can't remember all of them either, and I don't expect you to remember these structures. But you have hydrogen bond donors and acceptors which can now interact with base pairs in double-stranded DNA. These are all hydrogen bond interactions. Hydrogen bond interactions are weak interactions. If you think about interactions with methyl groups here, these are going to be your van der Waals interactions or some of the hydrophobic interactions. They're all weak interactions. So it's lots and lots of weak interactions that are going to lead to the binding of your amino acid, and it's not just one amino acid, it's lots of amino acids, to your particular double-stranded DNA. And in the book, they say this is an unpredictable interaction. So if you have a sequence of a protein, you have a sequence of a DNA, you don't know that one particular sequence of a protein is going to interact with one particular sequence on DNA. That's not completely true. People are getting much better at designing proteins that will bind to specific sequences. It's still not great, but they're getting a lot better at it. And partly why this is so challenging is that it's not just one amino acid interacting with one part of one base pair. It's lots of amino acids interacting with lots of different parts of lots of different base pairs in a number of different proteins. So here's just an example where we have one protein. It's in fact a homeodomain protein here that interacts with backbone phosphates with multiple different amino acids. Some of the bases are interacted with by single amino acids. Some are interacted with multiple amino acids. So there are lots of different interactions which are going on here. And I don't expect you to remember all of them. Again, scientists haven't even figured out how these guys are going to be interacting with each other either. So how do you get these amino acids interacting with things in the major groove? Well, not surprisingly, we talked about this already, it's all about alpha helices sticking down in the major groove DNA, or as we'll see a little bit later, a couple of beta strands. Why is that? Because, of course, alpha helices stick their side chains out on the outside, and the alpha helix, strangely enough, and again, almost certainly this is an evolutionary aspect of things, happens to fit perfectly into the major groove of DNA, and on the outside have these side chains which are going to be interacting with all the base pairs in the major groove of DNA. We'll talk about a couple of different DNA binding motifs. The most common one that you see in bacteria is the so-called helix-turn-helix motif. Again, what does it look like? Two helices, an N-terminal helix in blue, C-terminal helix in red. Why do people talk so much about this? Because these were the very first of these DNA binding motifs to be found. They're almost always present in dimers, which we'll talk about in just a second. But in the actual proteins, these alpha helices are not at all surprisingly about 3.4 nanometers apart from each other. How many base pairs are they apart if it's 3.4 nanometers? 10 base pairs. They're going to be on the same side of DNA. So each of these four very different gene regulatory proteins here binds to DNA, one turn of the helix apart. One of the monomers is here, the other monomer is here. So these are two separate polypeptides that will now bind to adjacent major grooves in the DNA. So binding here with our major groove and would be here with our major groove if the major and minor grooves are right on this structure, which they aren't. So why dimers? Well, the basic reason for dimers is that most of these alpha helices, if you look at that alpha helix, you're only going to be able to cover about 4, maybe as many as 5, in extreme cases 6 different base pairs. Even if we had 6 base pairs spread out through the whole genome, just at random you're going to have a sequence of those 6 bases very frequently in the genome. However, very rarely are you going to have a sequence of, in this case, 5 base pairs separated from the same 5 base pairs, and in this case is an inverted repeat structure because From 5' to 3' on one strand, we're going AACAC. On the other strand, we've got AACAC. Inverted repeat structure, one of your monomers is going to bind on one side. The other monomer is going to bind, here we are, right way around on the other side. That gives you a dimer. Now you've got 10 bases right next to each other. And again, this is just how many bases to the nth power, how often you're going to find these in the genome. This stretch of sequence you're going to find once or very few times in a genome on average, as opposed to this sequence, which you could find quite a lot. This particular example is the lambda CRO protein, control of repressor and other proteins. Very often people will talk about half sites. So one monomer is going to bind to 5' AACAC. So of course now it could bind to another half site AACAC. These particular dimers form this interaction here, supposed to be non-sequence specific. Each of these hexagons, or sort of hexagons here, is supposed to be showing that the protein is binding to the other half of the protein, which is in here. It's not just bacteria that have helices that get stuck in the major groove of DNA. This is sort of the, I like to call this the eukaryotic equivalent of a helix turn helix. I'm probably the only person in the world who actually uses this term, so don't worry too much about it. But basically, we've got a helix sitting down here in the major groove, and the helix right before it with another turn that goes into this helix. The big difference here is that we have a third helix, and this third helix should have some interactions in the minor groove of DNA. So it's not just major groove interactions that you have for these particular proteins. Mostly interacting in the major groove, but a few things here in the minor groove. Why homeodomains? Homeodomains turn out to be very common domains. Remember, this is domain versus a motif. Oh, wait a minute. Okay, what's the difference between a motif and a domain? So motifs are sequences but don't have a particular structure. A homeodomain now does have a structure. So those two helices by themselves, the helix turn helix, that doesn't have structure by itself. You only see it in the context of much bigger proteins here. On the other hand, the homeodomain has got structure and actually can bind just by itself. It's usually part of another protein, but can actually have this activity by itself. So that's sort of the eukaryotic equivalent, as it were, of the helix turn helix. Whether related to each other, people argue about. There's another kind of way that eukaryotes put alpha-helices into the major groove of DNA in terms of binding. These are what are called zinc fingers. Zinc fingers are only called zinc fingers because when people originally looked at them, they saw this extension here between either cysteines or histidines, which would associate together and give you a loop between these particular histines and cystidines. One of the really nice things about these particular structures is if you're just looking at an amino acid sequence and you see cysteine, 2 amino acids followed by a cysteine, 10 odd amino acids later, histidine followed by a histidine, or cysteine, 2 amino acids cysteine, 10 odd amino acids cysteine, 2 amino acids cysteine. Just by looking at this sequence, you can make a really strong prediction that this is going to be forming this kind of leucine zipper structure. Oh, no, leucine zipper structure. See? Got myself ahead already. Zinc finger structure. One of the best questions I ever saw in an exam was, can a zinc finger get stuck in a leucine zipper? That was from my undergraduate biology class. So, wonderful question. I keep telling the instructor whenever I see her. Great question. You can think about it. Great question. Yeah?
So what's the function of the zinc finger then?
So where you're getting here, it's not getting stuck in leucine zippers. But no, so what it does is this zinc finger coordinates these different amino acids in order to get an alpha helix positioned in such a way that it can bind nicely to DNA. And there are two different kinds of these. We've got those that have 2 beta strands with an alpha helix or 2 alpha helices with this zinc. Now here the zinc is this gray ball right here. But again, it's putting an alpha helix in the right place to bind to DNA.
So is it acting kind of like a glue to hold a particular portion of the protein together in a certain way so that it fits?
So you can certainly think about thinking of the zinc as literally glue, holding 2 different secondary structures together in order to get a potential DNA binding. And again, it's a motif because we're just talking about these sequences. It's not stable in and of itself. It's going to be part of a domain that's going to be binding to DNA. Yeah?
This whole idea, is it anything similar to how histones wrap DNA for condensing it?
So the question here has to do with, is this at all similar to how histones are wrapping DNA in order to condense them? Here we're talking about sequence-specific interactions with the DNA. So it's going to be a literal base pair that you have each of the side chains sticking out from these alpha helices that are interacting with. And so that's going to be quite different than what you see in histones, which are non-sequence specific. They're going to bind to any DNA. So the interactions are going to be much more with the backbone, the charged particles, or sorry, the charge interactions with their phosphates rather than the specific interactions with the base pairs. It's not just alpha helices. This is a really nice example of the MET repressor. We'll talk much more about gene regulation next time in bacteria. Originally, when people solved the structure of this DNA binding protein, they knew that it was binding to DNA. It had these 2 great alpha helices that were exactly the right distance apart to fit into the major groove of DNA. They were totally wrong. It's these 2 beta strands over here which interact in the major groove of DNA. 2 strands will fit in there, just like you have with alpha helices that are sticking the amino acids around the outside. With the beta strands, you're sticking up and down. And so, again, you can have those interactions directly with the base pairs from these 2 different strands. So here's the, and it turns out that there are other proteins that do this as well. P53, which is mutated in something like 50% of human cancers, also binds to DNA through these beta strand interactions. So it's not all about the alpha helices. Mass majority are alpha helices, but again, there are a few of these beta strand examples as well. So we talked about the DNA binding motifs. Now let's talk about dimerization motifs, because a lot of these are going to bind to dimers. And again, it gives you the specificity. You've got 2 half sites right next to each other. Dimerization. How are you getting 2 different proteins to come together? Well, classic example of that is a leucine zipper. But the leucine zipper here is, again, it's every 7 amino acids, large hydrophobic, will interact kind of like a zipper between the 2 different forms here. But a leucine zipper, in and of itself, is just dimerization. It's not binding DNA. Because, leucine, large hydrophobic, what large hydrophobic are you talking about in terms of DNA interactions here? So what that leucine zipper does is it brings 2 DNA binding either motifs or domains next to each other so that they can bind to DNA. And in this example, they're what are called basic helices because they have large numbers of positively charged amino acids which are going to interact with the DNA down here. Another very analogous structure in terms of getting dimers to form are these helix loop helix structures. Again, this here is the dimerization domain. This here is your motif, very often, which is binding to DNA. So it's between the 2 of them, you've got dimerization and DNA binding, dimerization and DNA binding. Just by themselves, a leucine zipper does not bind to DNA. Many people make that mistake. Leucine zipper is just coiled coil wrapping around each other, binding a dimerization. So why is dimerization so cool, flexible, great, wonderful, whatever kind of superlative you want to think about? We already talked about the fact that you get the specificity, but even if you've got two of exactly the same protein binding to exactly the same DNA, you put two of them together, you can get much higher specificity. But one of the great things about dimerization is, since they're different polypeptides, you can mix and match these. So you can have one of the monomers that binds to one sequence and a different monomer, as long as those two proteins are interacting with each other through their dimerization motifs, that could bind to a completely different sequence. So if, for instance, you have leucine zippers, which are just, again, every 7 amino acids, you're going to have a large hydrophobic, and then the DNA binding motif next to that could bind to separate sequences, you can have just two different leucine zipper containing DNA binding proteins that can bind now to 3 different sequences. The dark green binds to blue, light green binds to red, and the heterodimer, i.e. 2 different ones that come together, is now binding to blue-red. So 2 different proteins which can now bind to 3 different sequences, and all highly specifically because they've all binding to 2 half sites here. So this whole idea of mixing and matching proteins in terms of what sequences you can bind to, etc., is called combinatorial control, where you have expression of one protein, expression of the other protein, higher amounts, lower amounts, etc., of these all kinds of different proteins. Yes?
Shouldn't it be 4 sites? Could you do the other order?
So the question here is shouldn't it be 4 sites so you have red-blue here as well? DNA is symmetrical. So flopping it over the other side is just going to be putting these guys down here at the bottom. Now, as soon as you add a third, then, of course, you start to get different orientations relative to each other. But if we're just talking about 2, you're only going to have those 3 possibilities. Yeah?
So I'm just guessing in terms of regulation, the cell is able to sort of determine what combination it wants at a particular point in time to elicit a specific response. And so there's like signaling and all that stuff that happens to switch.
Yeah, so you're exactly right, and I'll paraphrase the question, that it's some kind of cellular signal which leads to production of one or the other of these, and actually you can control whether you form homodimers or heterodimers relative to each other. A lot of time it has to do with the other binding proteins or other DNA binding sites which are next to them. Yeah, there are lots of ways to control this. It's not just happening willy-nilly. Okay, and here are just a couple of examples of this. Here's the MAT-A1, MAT-alpha-2 that we're going to talk a lot more about when we talk about the actual regulation of certain genes here. Now, again, 2 very different proteins. This one over here and this one over here. You can see the structures are very different. Still, they've got alpha-helices that are binding in the major group of DNA. One of the other important things about this particular structure, you'll see the DNA is bent. And we'll see that turns out to be rather important thinking about nucleosome structures and DNA binding. Sometimes you even will have multiple different domains in the same protein. And so this is a particular example of a homeodomain containing protein and a POW domain containing protein. Again, the important names here are not important, but we've got 2 different kinds of DNA binding providing your higher specificity. And in some cases, these are all part of the same protein. In other cases, they're parts of different proteins. So let's look at a little bit more detail at how some of these proteins are interacting with the DNA. We already talked a little bit about consensus sequences before. This is the consensus sequence that one monomer of the nanog protein interacts with. Nanog actually has to do with the small crania that form when you mutate this gene. Turns out this is one of the 4 genes that if you express in an embryonic stem cell can cause it to dedifferentiate. So really fascinating protein, very important for regulation of developmental processes. So what people have found is they've looked at where this particular protein, again, it's very important for development, binds to DNA. And what they found is that most of the sequences that it binds to will have a T at the first position and of course an A on the opposite strand, because binding to double-stranded DNA, they always, every single sequence that this protein binds to has an AT base pair at the second and third position, but about 70% of the time it's got a T at this position, etc. And just like when we talked about the consensus sequences for promoters, anytime that this sequence is the consensus or very similar to the consensus sequence, this protein is going to bind very tightly. It's going to have a very low dissociation constant. Anytime it's very different, so say you've got a CAA, GAG, GC sequence, it's not going to bind as well there. And it's not going to be bound as frequently to that one particular site in DNA. And that seems to be one of the ways that the cell has evolved to have some of these gene regulatory proteins binding a lot in some places and some binding a lot less in other places. It's just how well it binds to any of these particular sequences. And so here is just another example of your homodimers, two dimers the same binding to one sequence, homodimers binding to another sequence, and then heterodimerization, which is happening. So that process leads to this concept of cooperative binding, and that goes back to thinking about how these dimers themselves are forming. How do you have each of the individual monomers interacting with each other? If these individual monomers are very tightly associated with each other, that basically means that your dimer is going to basically be acting like a monomer. So as you increase the concentration, you're going to get a little bit of binding and more and more and more. This is a classic kind of binding kinetics that hopefully you've seen in chemistry or will see in biochemistry, as the case may be. Increase in protein concentration is a slow and gradual increase, a little bit more at the beginning, a little bit later at the end. However, if you have mostly monomers floating around in solution and only occasional dimers, and it's those dimers that bind to the DNA, or even on the DNA you have dimer formation, what's going to happen is at low protein concentrations, these guys are never going to see each other. They're never going to come together. However, once you get past a certain point, now you'll have dimer formations, and those are going to bind straight to the DNA. And so this process, what it does is it basically shifts this curve from a gradual increase to an almost off or on situation. And so this is really sort of the idea of the switch, your cooperative binding here. When you have cooperation between this guy helping this guy bind, then you can have a much narrower window of protein concentration between, hello? Try that again. We're not over yet. Still got 5 minutes to go. Between being either off, i.e. not bound to DNA, or being on and being bound to DNA. So turns out this happens a lot with lots of different DNA binding proteins. And we already talked a little bit about cooperativity when we talked about binding of single-stranded DNA binding protein to DNA. Rec-A is also very much the same way, is that there's lots of monomers floating around here. As soon as one of those binds, you have a whole bunch more that bind. And that has to do with the interaction between the different monomer subunits. So I want to spend the last couple of minutes talking about nucleosome breathing. So how do you get binding by your specific gene regulatory protein to eukaryotic DNA, which is all wrapped up in nucleosomes? Usually nucleosomes, again, they're non-specific interactions, but they're very tightly wrapped. And very often, the major groove of DNA is going to be right next to your nucleosome. So it's going to be really hard to get your alpha-helices, your couple of beta strands in there. So one of the questions is, well, how can you get binding at all? Well, we talked a little bit about nucleosome breathing when we talked about these nucleosome structures way back when, like before the first midterm, like different worlds. But here, it's that you have sequences in your DNA that are going to be associated with the nucleosome most of the time, but some of the time it pops off. And what that happens is only about 1/20th of the time. But if that happens, and you happen to have a DNA binding protein around, it can bind to this sequence, even if that would be the major groove that's right up against the nucleosome here, but about 20 times less affinity, less of the time than you would have with DNA which is not bound to nucleosomes. Not surprisingly, DNA that's in the middle of the nucleosomes doesn't come off as much, and so it's also much lower frequency, but still a not unconsiderable frequency that you'll see binding to a sequence-specific DNA binding protein to this sequence here. Now, how do you get and how do you know that you've got this particular sequence right in the middle of the nucleosome? Because nucleosomes are binding non-specifically, right? Well, if you have nucleosomes that are binding to DNA that likes to be bent, then your nucleosomes are going to form specifically around those sequences. We talked about the AT bases, where you're getting squished on one side, and GC when they're open on the outside. So you can get sequence-specific nucleosome interactions and see where they're binding, and then see where your sequence is that you want them to bind to here in the first place. Another thing that we see is that if you've got a sequence here that one of your DNA binding proteins wants to interact with, if it's next to a sequence that's outside the nucleosome, just binding of one of these proteins very often can help binding of that second protein. And if you remember when we talked about MAD alpha, MAD A1, you saw the DNA was bent between those two monomers and they're bound to DNA. Well, that bending of the DNA is specific to those two, and it's not the same as the bending that you have around your nucleosome. And so that binding of those two proteins actually blocks nucleosome formation or may push it along a little bit. Many cases you also will have interactions between some of these DNA binding proteins and our friends the chromosome remodeling complexes, and so those will also help get the binding taking place here. So any questions about nucleosomes, DNA binding, etc.? In which case, you're welcome to leave, but I think this is an absolutely hilarious parody of All About the Base, and we'll see how this ends up working.